Well, thank you all for coming. I'm not sure where the best place to stand would be. Here, sound is good? Okay. So my research program in my lab, the students and I typically study chickadees and titmice in field settings. We do a lot of field experiments. And a few years back, I wanted to develop a new project that we could use in the lab. And so tried some things with hermit crabs, tried some things with pill bugs, and then tried some things with a species of cockroach that stuck. And this one is working and is really interesting in a lot of different ways. And so that's what I'd like to just introduce to you today. So why cockroaches? You may know cockroaches from things you might have read. So the famous Kafka story, Metamorphosis. Ian McEwan has a recent short book about not a human magically turning into a cockroach, but a cockroach magically turning into the Prime Minister of England. Just came out. It's maybe politically kind of relevant now. So why I'm bringing all this up is we typically view cockroaches as the ones we know about here, right? They're pest species, the ones that we come into contact with, the American cockroach, the German cockroach. And so our sense of cockroaches is one of revulsion, right? And I'll flip this because we're having lunch. But the fact is, less than 1% of the world's cockroaches are pest species in any way. So it's a very, very small percentage. And the vast majority of them you never see because they're underground or they're under the bark of trees. And if you live in the tropics or you visit the tropics, the ones that you're liable to see are often quite beautiful and really interesting looking. So this is a cockroach that has somewhat of the body shape of a pill bug, a roly-poly. It's much larger. There are cockroaches with really beautiful coloration patterns. This one is one of my favorites. I think it's from Guyana that almost looks like a moth. It's active during the daytime. It behaves a lot like a moth. And so I just want to give you the sense, hopefully by the end of the talk, you'll have a greater appreciation, if not a little bit of like fondness for this really interesting group of species. So cockroaches, I'm still learning. I'm not an entomologist, so I'm still learning like the natural history and the evolutionary relationships of cockroaches. But they're an incredibly diverse group of species. The phylogenetic tree that includes cockroaches also includes termites. The closest sort of sister group to this whole group are the mantids, so like praying mantises. So they're in a really interesting evolutionary place. The ones we're familiar with are the German cockroach, is one of the pest species around here. It's from the Ectobiidae. And then the American cockroach, which is a member of the Blattidae. The species I'm going to talk about today are a member of the Blaberidae family, and they're all part of this order of cockroaches that includes termites. It's a really diverse group of species. They have a lot of interesting behavior. I'm an animal behavior researcher. We've done some anti-predator behavior work with the birds that we study. Cockroaches have an enormous diversity of anti-predatory behaviors, from the kind of pill bug sort of rolling up into a defensive posture when predator stimuli are detected. Species that have aposematic coloring, so like warning coloration that can keep predators away. These two species have a behavior sort of like the bombardier beetle, where they can shoot out this sort of noxious, really distasteful chemical and sort of direct it towards a predator that might be bothering them. One of the things I'm really interested in is a lot of cockroach species actually have parental care, if you can imagine. A number of cockroach species are live bearing. This is a hissing cockroach giving birth to young. Once they give birth, the species that we study, the females tend to hang around the young for a very long period of time. In some species, like this species from India, the young actually climb on the mom's back and her wings cover them in a protective way, and she can move around with them in a secure way. And so there's lots of interesting behavior in this group of species. And so we'll get into some of the work that we've done. The other maybe odd thing about the talk, if you know any psychology, is why I would phrase social psychology with cockroaches. And so I want to talk a little bit about that. Just thinking very broadly about the social psychology of non-human animals. My own background is in, like Dr. Littman mentioned, in ethology. And ethology historically was thought to begin with Carl von Frisch and Konrad Lorenz. And then one of my heroes in science, Nico Tinbergen, was a Dutch early ethologist. And he wrote a paper in the 1960s that was sort of the basis for thinking about the study of animal behavior, including human behavior. And he basically argued that for any behavior that you might be interested in, there's 4 questions you can ask about it. 2 of them are what are called ultimate questions, and then 2 are what are called proximate questions. Ultimate questions are kind of at the level of the population or the species. Proximate questions are at the level of the individual. And so the 2 ultimate questions include phylogeny, so like the phylogenetic tree, the evolutionary tree I showed earlier. It's the evolutionary relationships of organisms. Those can sometimes tell us something about the behavior we might expect to see. Adaptation, so what's the current function of a behavior? How does it relate to fitness in individuals? And then the proximate questions are developmental questions. So what's the history of experience in that individual that led up to this point where it produces this behavior? And then mechanism, like what was the immediate trigger of the behavior? Tinbergen studied stickleback fish, and so these red-colored males, during the breeding season, when they see the color red, even if it's a fire truck going by the lab window, they start displaying aggressively. Red color is a trigger for their behavior. And so since I've been in graduate school, been thinking a lot about trying to combine these levels of analyses in the kinds of studies that I do. And social psychology as a field is currently, it's a field directed towards understanding human behavior, but it's a field that is interested in the notion of your current social environment has a powerful influence on your behavior. And so we look around the room, there's a lot of diversity. We differ from one another in lots of different dimensions. We've got different genetic backgrounds. We've got different experiential backgrounds, different personalities, different temperaments. All of that's important to behavior. But what social psychology says is that what we're doing here and now in this context also has a powerful influence on behavior. I wouldn't be acting like this right now if the room were empty. You'd want to see some professional help, get me some professional help if I were doing that. And you're behaving really differently right now than you would in a different kind of social context, right? And I've always been interested in social psychology because it really is a kind of integration of these 2 different levels of analysis, like the adaptiveness of behavior that functions really well in a particular context, and then the immediate social environment being a kind of mechanism predicting behavior. And going deep into history, what's interesting to me is that social psychology was not always having this really narrow focus on human behavior. The first handbook of social psychology that came out in 1935 has a number of major figures in social psychology. So Gordon Allport, for example. But if you look at the table of contents, there's some really interesting chapters that show up in this book. So social origins and processes among plants, insect societies, bird societies. Shelderup Ebbe did a chapter on social behavior of birds. This is where the first experiments on dominance hierarchies in stable groups of birds were discovered. So at the beginning of social psychology, it incorporated non-human animals as well as some non-animal systems. And so I'm the more I'm reading and thinking about these sorts of issues, I think we as animal behavior researchers could probably learn a lot by incorporating some social psychology principles, and social psychologists in turn I think could learn a lot by thinking about the kinds of things animal behavior researchers have been studying. And so just a really quick overview of some of this work. Katie Slocum and Klaus Zuberbuehler did this really nice study that came out about 10 years ago where a chimpanzee, if it's attacked by a group member, it typically produces like a call, a fear call, an alarm call of some kind. But it's got control over whether it does that. And it's much more likely to give that kind of a call if a more dominant individual is in the immediate social environment. So an individual that might do something to the attacker than if there's no more dominant individual in the immediate environment. In lizards, individuals do these really interesting dewlap displays and head bob and push-up displays. They don't just automatically do those, though. They sometimes do the display, sometimes don't. They change the rate of the display depending on the nature of their social environment, like how many males are in the environment, how many females are in the environment. They're really sensitive to the current social setting. And so there's this huge array of different phenomena that are studied in animal behavior that relate to this idea of social context being important. So if you've come across in things that you've read on audience effects, facilitation of behavior, stimulus enhancement, synchronization, cooperation, conformity, coordination, imitation, these are all phenomena that relate to the immediate social context influencing your behavior. And I've talked here to this group before about some of the songbird work that we do. A lot of that work has this kind of social context perspective. So the Carolina chickadee is one of the major species that we've been studying. And here's just a couple of the main data graphs from a recent study that Brittany Coppinger was the lead author on. She's a PhD student, where she set up aviary flocks of chickadees that all had exactly the same flock size. It's 4 individuals in each experimental flock. Half of the flocks were birds that were captured at the same field site, and so presumably are from the same natural flock. And then the other half of the experimental flocks were birds that were captured from different field sites, and so probably had not interacted with one another before. And we presented a number of different kinds of stimuli to them, control stimuli and novel stimuli, and recorded the latency with which they responded with a recruitment kind of call, an alarm kind of call. And we also presented predator-related stimuli, so like the model of an owl or sounds of predators, cat model. And what we found is that birds respond to these stimuli pretty quickly if they're control or novel stimuli, but if it's a predator stimuli, so this is a dangerous context, birds in the unfamiliar condition take a lot longer to produce that first call. And so an interpretation of this is if they're not among individuals that they're familiar with, that they've maybe reciprocated with in the past, I'm not going to call. I'm not going to draw attention to myself. This is a risky context. So that was one of the findings. We also found that just their normal call composition, the way they put the notes together to make this chickadee call that they produce, it differed based on whether they were in one of these familiar flocks versus one of these unfamiliar flocks. So the introductory notes that they build their calls with differed depending on which experimental flock they were in. So that's a quick overview of some of the social psychology of non-human animals and some of the work that we've done with birds. Let's do a quick overview of some of the work that's come out on cockroaches over the years. So this is the species some of you may be familiar with. It's the Madagascar hissing cockroach. I want to get some of these, but we don't have any yet. They get really large, like maybe that big. And they're called hissing cockroaches because they produce this audible sound, oftentimes when you pick them up. And people for a long time thought that this is an anti-predator response. It's like a startle kind of response. Maybe the predator drops you if they hear this hiss kind of sound. And so this researcher, Schotten, did this really simple study where she added males and females to different contexts that included only males, included only females, were mixed with predator stimuli. And what she found is that the one really strong social context that reliably predicts this hissing behavior is if you put females into these mixed groups. For whatever reason, they're much more likely to produce this particular sound. And so she thought this is maybe not an anti-predator behavior, maybe this is a conspecific sort of female recruitment kind of behavior. The German cockroach, this is one of our pest species around here. This research group did a series of studies looking at ingestive behavior and how that seems to depend on the social context. So they quantified how much food these guys are eating over the course of days of the study. And in this particular study, individuals were either isolated or in pairs. And what they found is that over the course of the experiment, paired individuals eat more per individual than the isolated individuals do. And this difference translates into different oocyte sizes. And so the oocyte is the cellular structure that produces eggs. The larger these things are, the larger the eggs that can be produced. So this is a fitness-related measure. And so individuals in the paired groups produced larger oocytes. And I apologize, I forget which one of these experiments did this particular manipulation. But what they found is that for paired females, the social housing condition, even if you control the amount of food, so they're not getting any more food than the isolate individuals are, they're still producing larger oocytes. There's something about being in a social setting that's beneficial to oocyte production. And then this group has done a series of these kinds of follow-up studies looking at the mechanism of all of this, where, again, this is oocyte length on the y-axis, isolated individuals, paired individuals of this species, they get the same effect. Much larger oocyte development in paired cockroaches. But then they looked at a lot of different sort of physical manipulations to see what are these animals really cueing in on as far as this social context goes. So just bumping into something in the environment, plastic beads, rubber beads, isn't enough to give you the effect. Having dead conspecifics in the cage, just the body of a conspecific, isn't enough. When you start getting into antennal contact and physical body of a living cockroach, that kind of stimulation is what seems to drive this effect in oocytes. Here's another example from a different cockroach species. So this is one of the classic ones that actually, it's one of the non-human examples that shows up in some social psychology and cognitive psychology textbooks. So Robert Zients did this series of pretty elegant studies of running roaches in different mazes. One called a runway design. It's just a single unidirectional tube. Another one in a maze design where they had to leave the area that's lit and go to a goal box at a right angle. And then there were conditions where inside these transparent, in this box that has these transparent tubes, there's also containers that are transparent that he can put in conspecifics, put in cockroaches as like observers watching the focal animals run. And so he ran these animals either as individuals or in pairs and found some pretty big differences in running behavior, maze running behavior. So pairs run faster than isolates in the runway condition, but isolates run faster than pairs in the maze condition. If you add an audience, so if you've got conspecifics in these open areas next to these tubes that these animals are running in, both isolates and pairs run slower, than in those coaction trials on the top. And then one of the additional manipulations involved a mirror task, where now the individual can potentially see a reflection of itself as it's running in the maze. In the mirror task, isolates run the maze faster than if there is no mirror. And then in some other conditions that involved chemical cues. So basically, they're really sensitive to the immediate social and in some cases physical context that they're in. That has a really powerful influence on their behavior. And then one last example is a study that is really interesting for the way the manipulation was done. It's a series of experiments that test roach shelter choice. So these are 2 shelters in a large test arena. This is a study of American cockroaches. So American cockroaches, German cockroaches, you may be familiar with this in your own living environment. You flip on the lights, you see a roach scurrying away into darkness. They don't like the light. They try to avoid being in the light. So they seek out shelter. And what these researchers did is, in some cases, vary the quality of the shelter by having some that are darker than others. And in this case, the first test I'm going to show, they had 2 identical shelters. So these are both kind of high quality shelters. And they had 2 conditions. One is just 16 live cockroaches, and they ask for these 2 identical shelters, which one do you pick? And they pick sort of randomly. Whichever one they find first, they hang out underneath, right? They're basically equally good shelters. Yeah?
Before you get too far from that previous slide that showed the mirrors and the audience, it seemed like the results were different, whether if the mirror, then the cockroach was seeing himself. In the audience, he was seeing others.
Mm-hmm.
Did I interpret that correctly? Is that
Yeah,
right?
yeah. So this is
an explanation for that?
No, and they didn't either in the paper. I mean, there's obviously when there's 2 roaches running in one of these confined spaces, there has to be some just physical impediment of sort of slowing down in some conditions. Maybe particularly in the maze condition. And so maybe that's part of the explanation. In the running condition where you have animals that are just sort of clustered but not necessarily running in this condition versus the mirror. The mirror, you're sort of seeing an animal running parallel to you. It's you, right? And so maybe that has a different kind of influence than just seeing a conspecific.
Roaches probably don't typically run into mirrors, so maybe they look in the mirror and say, man, that guy's ugly, I'm getting out of here.
Could be. Yeah, I don't know how good the visual acuity is of these guys, but they have pretty decent vision. And so, like exactly what they might be perceiving,
but they wouldn't know what they look like. So
they might know what a conspecific looks like, but just the anatomy.
It's another way they're going to try to outrun those.
Yeah, they wouldn't have a good sense of what they themselves looked like for sure.
Might notice that the mirror image was exactly mimicking
What they're doing. Yeah, so that might be an extra motivator to keep going faster. Yeah, so in this shelter design, what they found is that if it's just cockroaches, they pick randomly. In this other condition, they had 12 live cockroaches. These are the focal animals in the study, but then 4 robot cockroaches that were coded to find a shelter, but sort of just find the first shelter, pick randomly. And then if you look at the data from those 12 cockroaches that made their decisions, they're also basically flipping a coin. It's more or less the first shelter they find, that's where they end up staying. In a second study in this particular article, they had 2 different quality shelters. So one is not as good as the other. It's lighter. It lets more light in. The darker shelter is better. If 16 cockroaches are allowed to choose in brown, they vastly prefer the darker shelter, right? This makes adaptive sense. But if they're in a group with 4 confederates, 4 robotic cockroaches that have been coded to pick the lighter shelter, these 12 individuals are much more likely to be found under that lighter shelter than under the darker shelter. And so there's this kind of social peer pressure, maybe this social push on your behavior that can lead you to maladaptive decisions in this particular experiment. So take-home message from that quick snapshot of some social psychology research on cockroaches is, just like us, just like a lot of non-human animal species, cockroaches seem to be really sensitive to their immediate social environment. And so that leads into some of the first experiments that we did with cockroaches. And the very first study that I did is based really heavily on a study that came out about 9 years ago from Stéphane Canonge and colleagues. This was done on American cockroaches. And what they did is, in one of these large testing arenas, like in the previous slide, they have 2 different shelters that the roaches can go under. In this study, the shelters were either relatively poor quality, they let a lot of light through, or they were really good quality. They were pretty dark. They created a very dark space that the roaches prefer. And they tested these roaches in either as individuals, so isolated individuals, versus individuals in groups that varied from 10 cockroaches up to 30 cockroaches. And they noted what's the proportion of roaches across all the different trials that they did that were found under a shelter period. And it was a much higher percentage if the roaches were in a group as opposed to just a single individual. So in other words, single individuals don't find shelter very reliably. Furthermore, they break down of the individuals that found a shelter, how many were found under the darker shelter in the black bars versus under the lighter, the poorer shelter in the white bars. And what you see is that the single individuals are basically, whichever shelter they find first, they're probably staying under. And it's sort of flipping a coin which one they find first. But individuals that are in the groups are much more likely to be found under the darker, better shelter. And so read this study and thought, we can test this and we can expand on this particular experiment. And so we did it in a different species called the Argentinian wood roach, Blaptica dubia. This is a species that is a real favorite of lizard hobbyists, people that keep lizards and then need to feed lizards, because these roaches are sort of big and bulky, lots of protein, lots of calories. But unlike the American cockroach, unlike the German cockroach, these guys are really easy to keep in captivity. They can't fly and they can't climb very well. So plastic containers with reasonably tight-fitting lids is all you really need. So ease of care and maintenance is a big plus for this species. They're also sexually dimorphic, so the males have full-body wings. The females have these kinds of vestigial wings and frankly are more sort of attractive looking. They vary in coloration from yellowish, orangish, bright colors against black background. And so we wanted to do a conceptual replication of the Canonge et al. Study, but we wanted to add some complications and extensions to it. So Canonge et al. Was only done with male roaches. We tested both males and females, but in separate groups to see if there might be a sex difference in this shelter-seeking behavior. If you remember the graph, the groups in the Canonge et al. Study went from an isolated individual up to a kind of large group of 10. We had a smaller group of 5 individuals that we wanted to add. And then we wanted to add a question of cost, search cost. So we had a condition where the roaches could seek out these different shelters in a dark room versus in a room that's lit up, even brighter than the room we're in right now. And then we had a condition where the shelters are either far apart in the testing arena, so that's greater cost, or they're relatively close together, so that's lower cost. And each individual was tested under these two dimensions, so it's a 2x2 design. And so we also had the ability to test, like maybe these roaches get better over consecutive trials. Maybe by the 4th trial, they're figuring out the layout of this test arena and they're going to the dark shelter better. And then we had an additional experiment that I'll actually present the data for first, where we had a no-cost condition. So this is where the darker and the lighter shelter were actually touching one another. And so a roach could easily move underneath and figure out like which one is better. No cost of search. And so our design is pretty basic, again, because these guys can't climb to save their lives. So big plastic bins where we put the two shelters. Our shelters were made simply by cutting holes in these egg cartons. So this shelter lets in more light than this one. This one should be better. It's darker. It should be more preferred by roaches. And if you actually measure the light in the test arena and then under these two shelters, the darker shelter is significantly darker than the lighter shelter in terms of illuminance. And just for comparison, the Canonge et al. Study, these are the illuminance levels for their lighter, bad, darker, good shelters in those experiments. Yeah, and so this is what placement looks like if the video works. Oh, I don't have my glasses. Yeah, this is going to work. So we tried to release the roaches in a direction that was perpendicular to the shelters, so they would actually have to explore the space at least a little bit before they stumbled on a shelter. And so we left them in the test arena, this big plastic bin, for 2 or 3 hours in the light conditions, so where the lights are on in the room the whole time. We simply come in after this time period and then lift up the two shelters and count. Pretty straightforward. In the dark condition, we turned the lights on, counted 60 seconds, measured it on our watch, and then went in and counted the animals. This is counting data, and so the inter-observer reliability is really, this is really easy behavioral data to code. The one mistake was my fault. I counted wrong. Asan Akhtar, the student that was helping me, got it right. And so here are some of the data. So in the no-cost condition, so this is where these two shelters are touching. If they're picking shelters by chance, we should see roughly 50% of the time they're found under the darker, the better shelter, the good shelter. But that's not what we see. What we see is for the 24 sets that we ran, both females and males are selecting the darker shelter well above what we would expect by chance. No cost. If we factor in cost, so these are the conditions where the lights are on sometimes, shelters are farther apart from one another, what we find is that single individuals are picking shelters basically by chance. I think whatever one they find first, that's where they end up staying. But the three groups of roaches that were tested, they're picking above chance levels. They're picking the darker shelter. And furthermore, the larger groups, group of size 10 and 15, are picking the darker shelter significantly more often than the relatively small group. So I'll be honest, I was hoping we'd find some really interesting interaction terms and lots of cool sort of complications in the results. Nothing. There's no sex differences we could find. There's no cost of search differences that we could find. There's just this really large main effect of group size. We do know that the roaches, we didn't videotape these trials, and so we just set them up, we leave, we come back 2 or 3 hours later and count them. We don't know what they were doing during that time. But we do have a pretty strong reason to believe that they were searching, they were exploring the test arena, at least in the dark conditions. So if we just count up for all 24 groups, all group sizes, the number of times a roach was found outside a shelter when we went to count, in all 24 groups, we're more likely to find them in that lights off condition, right? So we flip the light on, wait 60 seconds, We're much more likely to find a roach outside of a shelter in that condition as opposed to the lights on condition. Yeah.
Is there any sort of leader, follower behavior?
It's a great question. We have no idea because we haven't videotaped, video recorded. That's one of the things we're going to start doing.
Put a colored dot on it.
Exactly. There's some software now that just by doing that kind of colored marking, you can code the computer to track these things over time. So we're looking into that. So the take-home message from this first experiment was we found a general replication of that Kananj et al. Earlier study. So there's a really strong preference for darker shelters when there's no cost at all to searching. But when there's any kind of a search cost, individuals are more likely to find that darker, better shelter if they're in a group than if they're by themselves. And so basically individuals can make more adaptive decisions when they're in a social group. And in fact, at least in this context, if they're in a larger social group, they may do better than if they're in a smaller social group. And so there's lots of follow-up studies we've got planned for this, but one that we went to immediately is the possibility of what's called conspecific cueing. This is seen in a lot of different animal species where individuals' habitat choice or shelter choice is based in part on cues of conspecifics, smells or sounds or sights of conspecific presence. And so we did the same basic shelter choice design for this first part of the study. We had two different sets of roaches that were 12 groups each. Each group we just randomly picked from our breeding colonies 2 adult females and 2 adult males and put them together into smaller shoebox-sized containers. And each one of those groups was a sort of independent replicate. We color marked individuals so we could get individual level data over time. And then we just kept these groups together for several weeks while we tested them. And in the first study that I'll talk about, the conspecific cueing study, we tested the groups with filter paper from either the breeding colony, so this is paper that just gets nasty with whatever chemical cues, the feces, the just cuticular hydrocarbon, sort of the byproducts of living that slough off on these filter papers, versus a blank filter paper right out of the box. And these are identical shelters in terms of darkness, so they should both be preferred by roaches that we're testing. And then a second study we did, unfortunately, only with the set B animals. I want to replicate this again with a larger set of individuals. When they were in the shoebox containers in their small groups, so 2 adult females, 2 adult males, we had filter paper that would come from their own group versus filter paper that came from a different group of 2 adult females and 2 adult males. And so what did we find? This is the combined data from those 2 sets of 12 groups each. And for both females and males, there's this hopefully really obvious preference for shelters that have the conspecific cues over the control filter paper condition. So identical shelters in terms of darkness, they strongly prefer the shelter that has the chemical sort of signature of this is another Blaptica dubia group. And so each one of these lines in this graph is a single individual's choice tested 6 times over the course of several weeks. The second study with a smaller data set, so these are data from just the set B cockroaches, where we test them with cues from their own group versus cues from the other group. We find the same significant effects, not as strong. There's more reversals here, but in both females and males, there's a significant effect for individuals to prefer the shelters that have their own group's cues over the cues of another group, a stranger group, if you will. And you may be able to get this from just sort of looking at these data in these graphs, but for whatever reason, males were found to be out in the testing arena, not under shelter, more often than females were across the 4 trials of testing that we did in this study. All right, so conspecific cueing for habitat choice, that's not that first really overwhelming finding. It's actually not very surprising because it's seen in a wide variety of species. This one is a lot more surprising, that they would have a preference for their own group's cues over those of another group. And why it's surprising is that these guys are not social termites, they're not social honeybees, right? They're not all genetic relatives. In fact, these are just unrelated females and males that were put together for this experiment. So we can't rely on those relatively simple mechanisms to explain the preference that they're showing. There is one earlier study that I've found in the literature that suggests this kind of, not so much a preference for your own group, but an avoidance response to cues of another group. So there's a little bit of a hint of this out there. But one thing that's interesting to me is that this suggests this is a learned behavior. They have to learn these cues to be able to show this kind of behavioral response. And so that leads into the future directions, and I'll try to wrap up here in the next couple minutes. Like, what could be explaining this own group preference that we found in that last experiment? So one possibility, doesn't seem very likely to me, but we'll throw it up there as a hypothesis, is that maybe over time, remember these groups they're unrelated individuals, but they're kept together for several weeks before they're tested here. Maybe over time their chemical cues, the composition of their exoskeleton is changed slightly. Maybe it's converged on a common chemical theme. That may sound outlandish, but this happens in human societies all the time with regard to dialects. So you move to a different area, there's processes like vocal accommodation and just learning the sort of components of a new dialect over time, right? That's vocal convergence. It's commonly seen in lots of species. So maybe there's convergence and then the learning of a group level cue. That's one possibility. Another possibility that I think is maybe a little more likely is every individual has, every human being has his or her own unique voice, different sort of visual gestures that they use, right? There's these own individual level signatures. It seems possible that each one of these individual roaches has its own individual level chemical cue characteristic. And then if that's the case, and you're in a group of these 2 adult females, 2 adult males for several weeks, you just have to learn 3 more, right, over the course of several weeks. That doesn't seem that difficult a task. And so maybe they're just learning each individual group member's idiosyncratic cues, and they're preferring those over stranger cues. Another possibility, this is the one that I'm thinking about testing first, is maybe all they're learning is their own cues. Maybe they've known their own cues growing up. They're continually surrounded by their own chemical cues. Maybe they learn what those are. And the filter paper that we used in that last experiment was housed in the container with not just the group members, but that focal individual. And so some of its own cues are getting on that filter paper. And so maybe that's all they're doing, is they're recognizing their own bodily cues and they're attracted to that and they're avoiding the strangers as much as possible. And I've just talked about all of this as if these are just cues. They're incidental sort of byproducts of metabolism and living, right? Feces, these hydrocarbons that are in their exoskeleton. But in a lot of animals, they rely on pheromones and they rely on vocal signals, so communicative acts that evolved for the purpose of communicating. And so it seems at least possible that some of these chemical characteristics may be signals rather than incidental byproducts of living. And then just to show you some pretty pictures, I'm trying to start testing more species now that I have the understanding that you can do massive studies in very small spaces if you work with cockroaches rather than songbirds. And so what we've recently gotten in the lab are 5 new species. Some of them have interesting names. The death's head cockroach, that sounds interesting just by how it's called. And so I'm starting breeding colonies with these 5 species. I'm going to try to get some others in. My only criterion is they can't fly and they can't climb, because I don't want to go through the extra mechanics of keeping hard to keep roaches in the lab. And other people in the Mossman building probably don't want exotic cockroaches running around their lab space. So with that, if I've got any time left, I'd be happy to take any questions. And I hope you have a greater appreciation for this wonderful group of species. Yep.
I'm just curious, are cockroaches cannibalistic?
Some species are. I'm trying to avoid those as well. I've noticed on the websites where they sell, there's a whole industry here if you're interested. There's people that seem to make their living breeding and selling exotic species of cockroaches. But they do mention that some of them you can't keep in too high densities, or you have to keep lots of food available, or they start to resort to cannibalism. As far as I know, these guys don't. Yep.
What kind of senses do they have? Can they smell? Can they hear? Like if you block their sense of smell somehow, would that interrupt their communication with each other?
So
Are they sending signals that they hear?
Yeah, my assumption was they're heavily in a chemical world, but they do have reasonably good vision. So they could be relying on visual cues. Like I would guess that even in the testing arenas where we ran these experiments, even in the shelters at a distance condition, so the high cost, I'd be surprised if a roach on one end of the container could not see a roach on the other end of the container. So just seeing another individual in another shelter, maybe that's enough motivation to go check out the other shelter. I'm teaching an animal behavior lab class this semester, and one of the groups is testing acoustic information. So basically they're playing back the sounds that humans make in kitchens, so like chopping vegetables and things like that, and seeing if that's an attractant for this species, Blapticadubia. And it turns out it is. They're attracted to the playback of this sound, and so they can clearly detect the acoustic information as well. They're covered in tibial spines, and my guess is they're really sensitive to vibrational cues. So like the footsteps of somebody approaching, even in the dark, might be enough for them to detect there's danger. So I think they're a lot more complicated in terms of their sensory abilities than we might give them credit for. I don't know.
Me first.
Great.
This may have been covered, but I'm a non-scientist, and maybe you covered it under cost, but they seem to be making decisions. And I know they make decisions for fear. Is it basically covered under kind of vulnerability feeling or visual? For light, dark, safe, unsafe?
Yeah, that's a great question. When I gave an earlier version of this talk, I didn't change the wording, so they're still referred to as good and bad shelters. They're good and bad in the sense of light and dark, but like your question is, if an individual is vulnerable, it's exposed in the open environment, this is where predators can get you easily. Any shelter is better than being out in the open. And so one way we can test that kind of question is by having the same shelter choice design, but then add like predator stimuli, obvious signals or cues of predators that might be in the environment. Yeah, so are they, if there's, I don't know, the smells of a cat or a lizard in the immediate environment, do they just go straight for the first shelter and stay no matter what, as opposed to a neutral chemical? Are they more likely to pick the darker shelter? That would be an easy experiment to do in the lab. Yeah.
On your shoebox experiments, for each experiment, do you use a virgin shoebox or do you clean them somehow? To reuse a shoebox, and if so, how are they cleaned?
Well, where they're housed for several weeks?
Well, housed or during the experiment.
Yeah, so after every trial, we wipe out the test arena with some like baby wipes to get rid of the cues. We don't want to use really harsh stuff like
Yeah.
bleach or vinegar or anything like that. Alcohol. But in the shoebox containers, we didn't clean them unless they got moldy. Then we'd clean them out and give them a shoebox container. But we wanted those cues to build up on the filter paper. So, yeah.
Yeah, given the small grain size, I'm speculating that there's a small number of algorithms and simple algorithms in play. Likely.
Yeah, I barely know the basic anatomy of a cockroach, let alone the nervous system, so I can't tell you anything about their nervous system. I'm really interested in the question of the possibility of really simple rules leading to complex behavior. That's seen in a lot of social species. And so I wouldn't be surprised if they're relying on, they just know their own chemical makeup. That's it, right? That could explain both of those. Well, it couldn't explain the first conspecific cueing one, but it could explain the own versus other group test. So maybe they don't have anything like group level recognition. Maybe it's just this sort of inherent individual recognition. But if you start thinking about that and you start thinking about, well, what does it actually mean to be able to recognize yourself? That leads into some really thorny consciousness, philosophy kinds of questions about awareness, right? Like self-awareness. This is, it's thought that, well, humans have it. Maybe some of the great ape non-human animals have it, but maybe dogs, maybe Alex, the African gray parrot, but not a whole lot of other species, right? But maybe cockroaches have self-awareness. I don't know. Something we could test.
Aside from placating your joy of playing with small insects like this, and what do you hope to kind of extrapolate? And is this something that you would hope that you would find some things that can be applied to humans that might guide things about human behavior, or what's the?
Yes, that's a great question too. Is the so. Part of the interest for me is getting back to, I used to be really good at math and really interested in math. And then for some stupid reason as an undergrad, I stopped taking math because I didn't need it for my major anymore. But there's lots of pretty basic analytical math models that can be applied to this kind of group level behavior, which again gets back at this idea of like simpler algorithms explaining complex behavior. So one of the things I would like to do is get to that point where I can start developing or collaborating with people that build these theoretical models of behavior and then test them experimentally. And then the broader question is the social psychology of cockroaches. I mean, are there some just universal social behavioral principles that operate in social species, whether it's a human community, it's a humpback whale, killer whale community, chimpanzee, cockroach species, that just something about conformity or approach behavior that is just so fundamental that it explains a lot of the behavior of different kinds of species.
Maybe one more question?
You have it.
Cockroaches are so elusive. How do you find this many cockroaches to stay?
eBay.
Oh yeah, eBay, Amazon. There's a great company called Cape Cod Roaches. This is where I got these species. There's a lot of labs that work on American and German cockroaches. You just catch them in urban environments and then start your breeding colonies. But yeah, I've been ordering them and then get them to breed in the lab. So
If people have additional questions, maybe Dr. Freeberg could stay for just a moment.
Yeah, I'd be happy to hang out.
And we'll look forward to his 4th talk at the UT Science Forum. Thank you so much.
Thank you all.